Greetings everyone. We would like to extend our thanks to Professor Anubha Gupta from IIITD Delhi and Professor Ritu from AIIMS Delhi for agreeing to give this interview. Welcome both. Ma'am, we would like to start with a small introduction of both of yourself.
Thank you, Abhishek. I am Anubha Gupta. I am currently working as a professor in the Department of Electronics and Communication Engineering at IIIT Delhi. I am also a member of Center of Excellence in Healthcare at IIIT Delhi. My current research interests are biomedical signal processing and image processing. I have a thriving collaboration with All India Institute of Medical Sciences, that is AIIMS New Delhi, from where Professor Ritu Gupta belongs. And we have been working actively since past 7 years in the area of cancer genomics and cancer imaging.
So hello Abhishek, hello Shubhi. Thank you for your time also. And I'm Dr. Ritu Gupta. I'm a hematopathologist and I'm a professor at the All India Institute of Medical Sciences, New Delhi. I'm interested in generating knowledge that improves our understanding of the process of oncogenesis. And I'm interested in developing the algorithms that are useful for early diagnosis, better prognostication and better management of blood cancer patients.
Can you throw some light on your journey from your college till today that can motivate our listeners? Also, what were the challenges you faced during your journey?
So I started my career with the Ministry of Information and Broadcasting, Government of India, as Assistant Director. I worked there for about 6 years, but because I was always more and more keen to learn, so I switched to academia. Then I worked as a faculty member at NSIT, which is now known as NSUT Delhi, for about 8 years. I got interested in higher education issues, particularly the retention of students and access as well as finance of higher education. So I moved to USA. I studied there, did my second master's, post-PhD in the area of higher education with concentration in higher education leadership and policy studies. Prior to that, I also did my PhD from IIT Delhi in 2006. So I was there in US for about 3 years at University of Maryland, College Park. I also worked as a senior administrator at one of the universities there, at Goyal State University. Post that, I came back to India and then I worked as IIIT Hyderabad as associate professor and eventually moved to IIIT Delhi in 2013 December. So if you see my career trajectory, I have worked in administrative positions, senior administrative positions, as well as a faculty, as a researcher. So I have enjoyed my journey throughout this time. So coming to the challenges, I would say everyone faces challenges. There are always ups and downs in career. So I have also faced many challenges and ups and downs in my career. But every time whenever I experienced any stumbling block in my career, I always introspected as to what was not working fine. So at times I took some steps backward, at times I really stepped out. But over years now when I reflect back, I really feel that those were small patches which disturbed me temporarily, but they also helped me learn and grow in my career. So I think I am very well satisfied as of today.
Well, my mother inspired me to become a doctor and she still inspires me. And she actually gave me a lot of strength, courage and inspiration to face any challenge that came my way during my journey of life. So professionally, I have enjoyed all the challenges that life threw at me. The only concern I have is that I missed a part of my son's childhood because of my work commitments. And in this, my family stood by me and supported me. So I feel that in India, we are blessed because we have a strong family culture that supports us, especially the working women.
So, as professors, your teaching experience must have been immense. So, how has been that experience over the years? And also, while interacting with students all over the world, what are the qualities you feel are present in them that are suited for research work, and what are the qualities which are not present?
So by now I have taught for more than 18 years, and I have taught at 3 different universities in India. That is at NSUT, IIIT Hyderabad, and now at IIIT Delhi. So the experience of teaching has been really wonderful, because all the 3 institutions, I would say, are the premier institutions of India, engineering universities of India. So the quality of students is really amazing and very good. So I have really enjoyed interacting with the students inside the classroom as well as outside the classroom. So when it comes to the challenges in teaching, I would say that over these years, technology has grown very rapidly. Perhaps this is the era when the technology is booming at the fastest pace. So this throws a lot of challenges for teachers as well as students both, because as teachers, we really want them to be skilled in the basics, that is the traditional basic engineering knowledge, as well as they should also be equipped with what's going on as of today in technology, in the industry and everywhere around. So but the duration of the degree program still remains the same. So that throws the challenge as to how to impart that education at the same time. So when I look at what the skills or the optimal skills that we feel are really important for students to be successful in their professional careers as well as in their degree programs, I feel that it is the theoretical knowledge, it is the coding skills, and these are the analytical skills which are on top of that are helpful for the students. So if I look at today's population, I find that our students are really comparatively much better in coding skills as compared to earlier days. They are more tuned to working in interdisciplinary teams of people, and perhaps they are more accommodating to people who are coming from different disciplines, because it's now the age where all interdisciplinary work is happening. I would say hats off to the boom which is happening in AI and related areas. At the same time, I do feel that our students over time, because of the availability of the material over internet, are becoming more accustomed to looking for solutions over the internet as compared to solving the problems on their own at the very first instance by hand, which we used to do. So I would say that it is slightly impacting their analytical performance when I look at the students whom I started teaching somewhere in 99 compared to the students that I teach today. So I think this is one aspect that we really need to work on. Perhaps the students as well as faculty both have to work on to improve this particular skill or strengthen this particular aspect. So because in the end, to be really successful in research, we really require all the 3, that is the coding, analytical skills, as well as their mathematical and theoretical knowledge.
So I would echo the thoughts and the views of Professor Anubha, and I would like to add that research per se does not have a format. It knows no boundaries, and it has a very diverse nature, and so is every student. Every student is unique, and as a mentor, we have to help them realize their own potential, and they can take it forward from there. So, some of the students are self-motivated, and with them, you just have to nudge them in the right direction. Whereas others, they require motivation, they need direction, and they need encouragement also. So I would say there is no particular format for research, there is no particular unique quality for research, but every student has a potential, and it is that variety of potential that makes the research work and the research work dynamic.
Thank you so much, ma'am, for your answers. So our next question is, can you briefly explain what your research was about? And also congratulations for your recent research paper publication.
Well, thank you, Shubhi. Actually, our recent research is on multiple myeloma, and multiple myeloma is a blood cancer in which primarily involves the bone marrow, and there is a transformation and proliferation of plasma cells. So currently, we have very effective treatments and very effective drugs against multiple myeloma, and initially 90 to 95% of our patients respond to the treatment. But eventually, all of them relapse. And this is the key problem which we would wanted to address. But to understand the factors that are responsible for relapse in this disease, we need to understand the pathogenesis of the disease and the clonal evolution or the evolution pattern of the disease. And for this purpose, in our joint collaborative effort with IIIT Delhi, we evaluated the tumor genomes of the myeloma patients.
So we analyzed the whole exome sequencing data, which is also called as the next generation sequencing data. So AIIMS collected, curated, annotated and gave us the data after due anonymity and all that stuff. So we employed AI and ML methodologies to process this data. So if I tell in brief what WES data is, this is actually a sequence of 4 nucleotides which are called as the A, G, C and T. Now the whole human genome sequence consists of this sequence of nucleotides, and there is a set pattern of nucleotides in particular genes which are located on the chromosomes. Now when this pattern is altered, it is also called as the mutations, and these mutations lead to the disruption or the malfunctioning of the genes, which ultimately impacts the human metabolism. So in this WES analysis, what we look for is that we collect the WES data of a particular patient, we match it with the referral human genome to find the mutations, then we find the genes that have got mutated, and eventually find which biological pathways these genes are part of and hence are affecting them in a altered manner. Now once this is found out, then we can find out that what is leading to the cancer genesis as well as progression, etc. So we analyze this WES data to find out the alterations or the mutations and finally the genes that would help us or help the doctors or assist the doctors in figuring out the reason for the progression of the disease.
That is certainly very illuminating. So, apart from you both and your teams of students, what roles have been played by the two collaborating institutions in this sort of research?
So, this is a very collaborative and interdisciplinary work, and also has a huge potential for translational, translation to actually therapeutics, personalized therapeutics. So as far as IIIT Delhi is concerned, we are more into development of methods or analysis of the data, innovating new methods if available tools are not sufficient to analyze the data. Whereas when it comes to AIIMS, they collect, curate and provide us all the inferences even during the research stage. So whenever we design a particular or implement a particular method, so whatever results are generated, we actually run through those results with the AIIMS team and they tell us whether the inferences are appropriate or correct or not, and should we do away with this particular method or should we be innovating a different method to have better results.
Well, I would call it a symbiotic relationship where the medical and the engineering communities come together to find a solution. So the innovative solutions are developed by them and they are used by us to evaluate the clinical data and to derive information of translational value. So this is how this symbiotic relationship works.
Can you briefly discuss your research and the key findings for the cancer research community?
So in the first work, we as we said that we are working on multiple myeloma. So multiple myeloma has a precursor benign stage which is called as the MGUS. So doctors are interested in knowing how this disease progresses from MGUS to MM, or what are the factors responsible for that. So in the first work, which was using AI and ML technologies, we analyzed the data, but one of the challenges that we face in this data is that the cancer is a heterogeneous disease. That is, it differs from patient to patient. The genomic profile and the mutations, they differ from patient to patient. At the same time, the data is also very large and humongous amount of data that needs to be analyzed. So since there is no real ground truth also known on the data, it's just the real data set that you need to analyze. So that means whatever inferences you or whatever results you extract out of the data, you really need to be really sure of that indeed these are the biomarkers or these are the reasons that are leading to progression of the cancer from one stage to another or within a cancer stage. So what we did was that we implemented both sorts of methods like pre-processing as well as later on AIML methods to infer the results out of the data. But what we did to have robust findings was that we utilized multiple tools and used the consensus of those even pre-processing tools to figure out the mutations, key mutations, and later on we implemented multiple classifiers also to infer out the results, so that there are robust findings and we are able to give the provide the genes which are mutated only in perhaps MGUS, only in MM, or both in MGUS as well as MM, to figure out what are the differentiating genes or mutations which are happening in the two population.
So that was the first part, and in the second part we looked at the longitudinal whole exome sequencing data from multiple myeloma patients. So in this data we found founder clones and multiple subclones, and we evaluated their trajectory over a period of time, and we had a couple of interesting findings. And the first very interesting finding was that we found a lot of interpatient as well as intrapatient tumor heterogeneity, which essentially means that a single patient had multiple tumor clones at the same time. So, it would be interesting to see how each of these clones respond to the individual drug that is given as a part of combination therapy to these myeloma patients. Then we also, when we evaluated the trajectory of these clones over time and after giving the initial therapy, we realized that these clones evolved, and there were three distinct patterns of clonal evolution. In the first linear pattern, we found that the clones progressively acquired more mutations. That was the simplest pattern. Then there was another pattern that was a stable pattern of evolution, where some of the clones were being lost and some of the mutations were being lost, but largely the tumor clone remained stable. And the third is the most interesting and the most frequent pattern, which is the branching pattern. In this, what happens is that from a single clone, some of the tumor cells acquire one set of mutations, whereas some other cells from the same clone acquire another set of mutations. So what we found was that this pattern was most frequent and was associated with patients who responded better to therapy. So we can say that this pattern is evident of clonal evolution under the stress of therapy. And another interesting finding was that, when we looked at the type of mutations that were gained or lost in these clones, we found that some of the mutations in the driver genes were either gained or lost, which essentially means that at different stages of the disease, different driver genes are playing a role. So different driver genes are modulating the tumor genome or the disease at different time points. And the changes, the loss and gain of the mutations in the actionable genes is actually clinically very important, because it essentially means that if we are thinking of starting a targeted therapy in a patient, That means we need to analyze his tumor genome just prior to the start of therapy to see whether this patient actually still possess the actionable mutation against the drug that we are planning to use on. Yeah. Then another interesting finding was that we found that some of the patients had very high load of mutations. Traditionally, we call these patients as hypermutators. So in some of the cancers, we have seen that the hypermutators respond best to immunotherapy. So perhaps in myeloma also we can selectively give immunotherapy, or we can select this group of patients to give for immunotherapy kind of a treatment. Then we also looked at the single base substitutions, and using these single base substitutions you derive something called the cosmic signatures. So we found that in our cohort the chemotherapy signatures and the DNA repair pathway signatures were associated with inferior outcomes. So the cosmic signatures for these two features can be used as biomarkers in myeloma patients in future.
So, although you are an engineer, you have conducted research on multiple myeloma cancer. So, how did you go about finding and approaching the patients and the hospitals, and were they willing to help you?
Yeah, so I would say there are two key ways in which particularly engineers are working in the biomedical areas. One is by utilizing the publicly available data sets, applying methods and drawing inferences out of those data sets. The other way is to directly work with the hospitals. So at least we at SBI Lab are directly working and collaborating with the hospitals. So AIIMS and IIITD being in the closer vicinity, it really makes them an ideal partner for us. Moreover, when it comes to AIIMS, we know that this is the best multidisciplinary hospital of the country, and perhaps the last medical referral spot for critical patients in the country. So the expertise of the doctors, the quality and the quantity of the data available with AIIMS really makes them an ideal research partner for us. So we have been collaborating with them since past 7 years, and it has been really a satisfying journey of working with them. Their insights on the data, their expertise, and the possibility of really putting it into the practice in therapy really is exciting for us also, and that motivates us to work more and more with them.
What were the challenges you faced while carrying out your research, and how did you address them?
So as far as data analysis is concerned, I would say that we faced a couple of challenges, and we are still facing them, particularly when it comes to biomedical areas. So the very first challenge that we always say is there is no ground truth. That is, you get the data of a patient and you have to figure out, like, innovate methods, devise new methods, or implement existing state-of-the-art methods to figure out what are the genes which are playing, working as, let's say, key biomarkers. But no one knows what are the genes which are actually playing key role in this particular data. So that is one of the bigger challenges, and how we counter that is that we make use of multiple data sets. That is, we are working utilizing the data set that is being provided by AIIMS, as well as we also look for the data sets which are available at public platforms or can be arranged through other platforms or media, or maybe through other hospitals, etc. So working with the different data sets of the same disorder really helps us validate the findings. So that is one way. The other is that we are also working with the workflow or the pipelines where we utilize multiple tools on the same data and then look for the consensus results. So of course there would be some differing results, but some common results, but it helps us figure out if we are if there are some errors in the processing of the data. So that is one. The second important challenge that is always there in medical data sets is generally they are small, in the sense that your data is not representative of the entire population. So let us say we are analyzing multiple myeloma data set, then the amount of data set that we would have will not be the representative of the entire multiple myeloma population. So that throws another challenge, because that implies that different methods would give different results, and even within the same data set on different patients also we may find different results. So and then whether the method or whatever pipeline we have deployed, is it really going to give robust results on the prospective patients' data? Now that's also very important. So I would say there, care needs to be taken by the engineers in innovating or devising methods, that they are able to bring interpretability in the methods, that so that you know for sure that indeed it is learning the desired features and is not doing something else. And also be in touch with the doctors at different stages of the processing so that they can help you be on the right path. The third challenge that we always face is the imbalanced nature of the data set. So always the disease population, or in general the disease population data, is always less as compared to the healthy population. When in this particular case, when we are looking at MGUS versus MM, because MGUS is a benign state, so for sure not many patients would be reporting, such patients would be reporting to the hospital. So we had an imbalance of roughly about 20:1, where MM population samples were very large.
So this research is certainly groundbreaking in the way that you are counting the challenges which will help the cancer patients extend the mortality. So how do you plan to extend this research further?
Well, multiple myeloma is a multi-gene disorder, and all the mutations put together have a cumulative impact on the transcriptome profile. And as a result, the different pathways that are deregulated in the process. So, we would like to decipher the entire tumor genome axis of multiple myeloma through multiomics analysis, through functional genomics, and through single cell transcriptomics. And another aspect that we came across in the study was significant, as I mentioned earlier, significant intra-patient tumoral heterogeneity. And we observe waxing and waning of clones and acquisition of new mutations. And therefore, we need to look at risk as well as time-adapted therapies in future for these patients.
So we are looking at deep learning and bio-inspired architectures to solve the problems that are thrown to us by the doctors. So these days, a lot of work is also being done in interpretable artificial intelligence, which is also called as explainable AI at times. So we would be utilizing as well as innovating interpretable AI solutions to assist the doctors in providing better treatment to cancer patients.
The amazing results that you and your students have achieved reflect how successful this collaboration has been between you and the students. Can you share with our viewers that how this all started and how did you ensure that there was no complication there?
So I would say that every discipline has its own language. So I must admit that it was not so easy for us to also come on the same page. So initially when we started interacting, it was like Professor Ritu wanted us to understand the biology of the disease, and we wanted her to understand how or what methods are being implemented and what those methods do. Now, it was really hard for both of us to understand that there is a limit to what we can explain to each other, in the sense that what can be assimilated and understood by the other person who is coming from a different discipline. So I would say that was a bigger challenge initially for both of us to counter. Now, there is another added challenge, because doctors believe in evidence. They happen to see patients, how it is translating. So they believe in evidence, whereas when it comes to engineers, we imagine. We then build hypotheses and then we innovate. So we are always in a different world, that we want to do something that has perhaps never been done before. So this was again a point of contention, that we needed to explain to each other that this is how she would say this is how we do research, and we would say no, this is how we do research. Something which is not done yet is a problem research statement for us. So I think it took us a while, but I am really glad that both of us understood over time that we need to understand each other as well as at the same time explain only the minimal required amount to each other of our own discipline so that it can be digested by the other person. But I think we are fortunate enough to be on board now.
I think Anubha has put it in a very positive fashion, and the initial phase was quite frustrating, believe me. So, finally we came to terms once we understood that we need to work on each other and on ourselves also to make ourselves understood by the other side. So for this purpose, we held a number of joint meetings on site as well as online. So how we overcame this challenge was that the students from both the sides, they presented papers. And while going through discussing those papers jointly, we tried to understand and convey the context, the fundamentals and the methods to each other. And that helped build our understanding of the concepts that are required to be understood before a research question can be taken further. And in addition, our students also visited each other's lab, and they also worked. My students worked in Dr. Anubha's lab and they worked in my lab to better understand and get the grasp of the subject.
So, Professor Ritu as head of laboratory oncology at AIIMS, and Professor Anubha as the head of SBI lab and chair Delhi chapter of IIITD signal processing. How do you think the synergy between institutions and international forums can help in advancing research to the next level?
So definitely synergy is required when you want to work in interdisciplinary areas. But I believe that such collaborating institutions can really do wonders. So for example, I can cite some of the ways that we have been working with. So we are jointly supervising PhD students and training them. That's one way to go about that. Then we are also, like, whatever methods, tools we develop, we are also releasing them at the public platforms. So if you visit my lab, many of the codes have been released by my students over GitHub, etc. Then we are also curating, AIIMS is collecting, curating, annotating data sets. We also support them at times in these tasks. And we have been releasing data sets also at the public platform. So far we have released 4 imaging data sets, cancer imaging data sets jointly at the public platforms. Then we have also hosted, organized 2 medical imaging challenges at international conference, that is IIIE ISB 2019 and IIIE ISB 2021. So I believe these are the ways that other students, researchers, they also get inspired, they get the head start on the data, as well as they get to know about the problems. So, and definitely being part of the IIIE Signal Processing Society also helps, because we really get networked, we really get forums to present, and we get to know what other teams and what other leading institutions are doing in their research domains.
I would say genesis of a good research question, and then testing and analyzing that hypothesis from multiple aspects and translating the knowledge generated into clinical practice are some key aspects of the healthcare research. And this becomes a complete circle. So one can enter the circle at any point. You may not start from the first part itself. You can enter the circle from any time, but to complete the circle, you require multidisciplinary skills. And for this, synergy and integration of the institutes harboring complementary skills is the key to complete the circle and to make a fruitful or applied solution in the healthcare sector.
Thank you so much, Prof. Anubha and Prof. Ritu, for taking out the precious time and for this amazing interview. It was very insightful for all of us. Thank you.
Thank you, Shubhi. Thank you, Abhishek. Thank you so much.
Thank you. And thank you, Dr. Anubha, for bearing with us and partnering with us.
Thank you so much. Thank you. Thank you, everyone.